So, beginning today we will look at a long series of videos wherein we will study constraint based modeling of metabolic networks. This happens to be one of the most important tools to study metabolic networks and a very favorite tool in systems biology, because it can make predictions about large genome scale metabolic networks and so on. So, today I will introduce you to constraint based modeling. What are all the constraints that pervade biological systems, and how do we compute what is known as the stoichiometric matrix, which is a nice representation of a metabolic network for further modeling. Welcome back. We will move over to the next module of the course, wherein we are going to talk about constraint based modeling. We have seen a few other paradigms for modeling, particularly network based modeling or network biology, followed by studying dynamic models with special emphasis on parameter estimation and so on. Today we will start looking at constraint based modeling, which happens to be one of the most powerful techniques to study biological systems, especially at a system scale. So, I think one thing that you may be convinced with already is that we need alternatives to kinetic modeling, right. So, why would we need alternatives to kinetic modeling? Essentially, but the key thing I want you to hit at right away is that kinetic models involve very large number of parameters. If you want to build a genome scale kinetic model, people have, by the way, it is very tricky, very difficult, simply because you have to estimate something like 2000 parameters. Unless you have very respectable tight estimates for each of the parameters a priori, you are not going to be able to estimate or build such models very effectively. But people are bravely attempting this, and with good reason, but clearly you need a simpler method, right. And we have already discussed at the very beginning, it all depends on the questions you want to ask. If all I want to know is whether post this gene deletion is my E. coli going to grow or not, I do not need to build a dynamic model. Most likely I do not need a dynamic model, and we will see how constraint based models can actually give you answers to questions like that. So, in constraint based modeling was literally founded by Paulson and colleagues. Paulson was at UCSD then, and I think he is one of the major proponents of constraint based modeling, and their lab has brought out a lot of work, and there are many other fantastic scientists who have since contributed, notably Kostas Maranas, Eitan Rupin, and a few others, many others, and there are other people like Nielsen and so on, who have made a lot of contributions to constraint based modeling in terms of building models and using them to make predictions and so on. So, what is constraint based modeling? What do we mean by constraints? I am just asking a generic question. I am not trying to motivate what I am preempt what I am going to cover today, but what do we mean by a constraint in general?
Limit some guidelines and limitations.
So, some limitations, I think limitation is a good word, right, and it also makes a lot of sense in the context of biological systems, because you always talk about a growth limiting substrate, right, something of that sort. So, there is always a constraint that every system has to function with, right. So, constraints govern all possible biological functions, and organisms exist in resource scarce environments, and fitter organisms survive with higher probability, and fitness itself requires satisfying several constraints which limits the range of observable phenotypes. So, at this point I want you to think about what kind of constraints do we need to satisfy to live, or what organisms need to satisfy to survive, what constraints must they satisfy. Can you think of some simple constraints?
The amount of nutrients one should need to concentration.
Yeah, so nutrients, but something more macroscopic.
Water, oxygen.
So essentially a lot of nutrients and input and so on, but other than that, there are fundamental constraints on even how big an organism can be. So, this is very classic essay which I like to point to at this point. A really decorated biologist, JBS Haldane, who essentially wrote this essay in 1926 I think on being the right size, where he essentially argues that a human being can only be so big, an elephant can only be so big, whereas an insect does not need to have oxygen in its bloodstream, because it has got such a small body to power it can take air by diffusion, take oxygen by diffusion, right. So, there are constraints. So, if you want to have a bigger animal, it has to have better machinery. So, why do not we have a 20-foot tall human? Could we have? Well, we could, but what would be the challenge?
Bones won't be able to support that.
Yes, in fact, that's actually what Haldane argues, right? He argues that you can't there is some dependency between the weight an organism can be and the cross section of its bones, right? Because it has to support that kind of a weight, right? But even simpler than that, I would think of something else before even I go to bones. Yeah. So, can you have a heart that's powerful enough to pump blood 20 feet in a human being? I mean, it does it in giraffes, but can it do it in human beings? Right. So, those might be the constraints. So, these are some sort of implicit constraints. You do not really stop and think about these constraints, but clearly there are some constraints that you have to follow. If you step to the microscopic level, there are clearly other constraints that one has to follow. For example, thermodynamics. No reaction in your body can violate thermodynamics. No reaction in your body can violate stoichiometry, right. So, there are some such obvious constraints that exist in nature. So, fitness requires satisfying several constraints which limits the range of observable phenotypes, and survival depends on the best utilization of resources to survive and grow, but always subject to constraints. You can never violate constraints. So, the next idea is that since phenotypes must satisfy constraints, constraints can help eliminate impossible behaviors, right. And this is where I borrow this classic slide from Paulson, who asks how often have I said to you that when you eliminated the impossible, whatever remains, however improbable, must be the truth. The famous fictitious character, right. I think you must be familiar with Sherlock Holmes, right. So, this is a very classic example on you basically start with a big pool of suspects and then you go about eliminating one by one to arrive at a smaller pool of suspects who have to be tested in a different way. We essentially do the same thing here. We start with an unbounded solution space, right. So, think as a space of possible solutions. But what do we mean by a flux distribution in the first place, right. I want you to recall what a metabolic network is. A metabolic network for our purposes is a long list of reactions. I said approximately this is what a metabolic network is. If you recall, we had an analogy with traffic networks and things like that and so on. So, this is your metabolic network, which can also be written in the form right. You remember we did this on how do we write out each fluxes. So, you would say that dA by dt is going to be minus VR1 or minus V1, right, and dB by dt would be minus V1 minus V2, right, something of that sort. So, now these are your rates which we also call fluxes. What is the flux in general? Chemical engineering?
Quantity per unit area.
A flow per unit area. Here we just drop the unit area. We just talk about flow or rate of any given chemical reaction. So, given this system, we are tasked with finding rates of all reactions, meaning V1, V2, Vn. This is what we call a flux distribution. Right. So, what are the fluxes of every individual reaction in a network? This essentially specifies a network for us. So, think of this as a flux space, right? It's a 3-dimensional space. So, you have flux A, flux B and flux C, right. So, just think of it as 3 reactions V1, V2, V3, right. Now, you apply constraints to eliminate improbable suspects or impossible, infeasible solutions, infeasible suspects or solutions, and you come up with a much smaller subset. So, you will note that from one infinite subset we have gone to another infinite subset, but clearly this is quite restricted. So, the idea is simple. What we do is, you may actually be very familiar with something of this sort. I will come back to it in a moment in a few slides. You start with, so now any point in this space, in fact I should draw it this way, any point in this space is a possible solution, right, for a system with 2 reactions. But now if I say V1 greater than 0, V2 greater than 0, it becomes this region. Now if I say V1 equal to V2, it becomes this very line, right. So, every solution on this is a possibility, right. And then if I know something more, if I know 0 less than V1 less than 5, right, that might say that all the solutions are on this part, let us say this is 5. So, you can, these constraints basically help eliminate impossible behaviors, impossible flux solutions. What kind of constraints do we put? How do we impose these constraints? Let us look at it. Right. So, you can think of different types of constraints. Some classic examples would be environmental constraints. I think that is something everybody thinks about first up. What are the nutrients available? What is the availability of my carbon, oxygen, sulfur, nitrogen and phosphate sources? Of course, pH, temperature, osmolarity and availability of NADH, NADPH and so on. And many of these are condition dependent constraints. So, they are actually variable, they are not fixed. Right. This is one set of constraints. The other really interesting set of constraints are, there are some self imposed restraints by the cell, which is almost counterintuitive, right. So, you expect that the cell will do something, the conditions are actually feasible for the cell to do something, but it actually does not. Why? Can you think of an example?
Surface area or volume.
What's
If it exceeds a certain number,
it has to?
divide this.
Well, so that could be one, yeah, right, because it's not something. So you think the cell can potentially survive with a larger volume or something, where it has to break down into 2. Yes. What else?
If you overproduce something inside, you might lose it by diffusion, maintaining ion concentrations.
But that's not a self-imposed restraint actually, right? What would be a self-imposed restraint? The cell can grow, but it may choose not to, for example. I think you're very familiar with two examples of this sort. When does the cell choose not to use a substrate? Catabolite repression, right? That will be a classic example. As lactose and glucose available to the cell should be growing, but it will just use should be growing at a higher rate using both glucose and lactose, but the cell will choose to use only glucose because there is a catabolite repression on the utilization of lactose. Once glucose is exhausted, you have a dioxic shift, and the lactose enzymes are then expressed, the genes are expressed, and then the lactose metabolism kicks in. Right? So the cell essentially chooses not to use lactose, right. So another very similar example, a cell chooses not to grow, sporulation, right. So the conditions seem to be reasonable for growth, but the cell is trying to eliminate a suboptimal phenotype which could be reasonable growth under some harsh conditions and confine itself to a behavior of increased fitness. It really depends upon what you define fitness, but if you talk about fitness as the ability to survive and live on for more generations, maybe the cell is achieving that by sporulating, right? So the spore is more stable, and when the conditions become more favorable, the cell can again start growing and so on, right. So there are such self imposed restraints as well, but we will focus on simpler constraints, essentially connectivity coming from system stoichiometry. First up, what is stoichiometry? In what ratios, right? So there are clear dependencies. Okay. So 1 mole of glucose will produce exactly 1 mole of fructose, not a molecule more, not a molecule less. 1 mole of glucose will produce exactly 2 moles of pyruvate via glycolysis, right. So this becomes a nice constraint, and of course there is thermodynamics. There are some reactions that can only go in a particular direction, some reactions would be reversible, they can go in either direction, but there are such constraints. Then there are maximum flux rates, right. So every enzyme has a particular turnover number, right. It may not be able to turn over more than x number of molecules per second, and there are there's genetic regulation, there's you need to worry about time constants, mass action, there are many other constraints that you may be able to think up of, but most importantly I would say are connectivity, thermodynamics and capacity. So what would this translate into practice? We will see shortly. So let us take a very simple example. What is the size of the stoichiometric matrix for this system? 5 cross 3. You have 5 metabolites and 3 reactions, and you have a 5 cross 3 system. So let us just go back to the concept of the stoichiometric matrix. What do you think? Do you usually have more metabolites than reactions, or more reactions than metabolites, or roughly the same number of metabolites and reactions? So are you going to have a square stoichiometric matrix, or a fat stoichiometric matrix, or a tall matrix? Tall, that would mean this. Why? right, but the same metabolite can also participate in more than one reaction, right? So you would see that ATP participates in 100 reactions, and maybe glucose participates in like 30-40 reactions, and there are other metabolites that will participate only in one or two reactions. You might see a sort of power law there as well normally, right? So you would normally observe that the same metabolites mix and match amongst different reactants, right? Some obviously some metabolites keep changing, so invariably you have M less than R, which means you have fewer rows than columns and you have a fat stoichiometric matrix. We will see what implications this has. So for now, you remember that you can set up the equations like dx by dt is S into V, right. So what are the V's again? Fluxes. What is X? Yeah, concentrations. Metabolite concentrations. The rate of change of metabolite concentrations linked via the stoichiometric matrix. So I hope in today's video you got an introduction to this interesting field of constraint based modeling, followed by a brief insight into the kind of constraints that exist in biological systems and how one goes about building the stoichiometric matrix. In the next video we will start looking at flux balance analysis, and we will start with a steady state mass balance, followed by how one adds constraints to the mass balance problem, and then how one sets up the optimization problem, which typically is a linear programming LP problem.